Hi, my name is Farah Zulaika binti. Today I will be talking about the introduction of titration. Titration is an analytical technique used to determine the concentration of an unknown solution by gradually adding a standardized solution, the titrant, until the reaction reaches its endpoint, which is indicated by a color change. The volume of titrant required to reach the endpoint is then used to calculate the concentration of the unknown solution. So, today, our group will conduct an experiment to determine the percent content of acetic acid in vinegar by titration. The objective of this experiment is to determine the concentration of acetic acid in a vinegar sample. Now we move to the theory. The principle of titration. Titration is a process where a solution of known concentration, the titrant, is added from a burette to another solution known as the analyte until the reaction between them is complete. There are several types of titration used in analytical chemistry, including acid-base titration, complexometric titration, precipitation titration, and redox titration. First, acid-base titration. This is the most common type of titration where an acid is titrated with a base or vice versa to determine the concentration of the unknown solution. Next, complexometric titration. This involves the formation of a stable complex between the analyte and a compexic agent such as EDTA. The endpoint is detected using a metal ion indicator. Next, precipitation titration. Here, the analyte is precipitated by the gradual addition of a titrant, and the endpoint is detected by monitoring the change in turbidity or using an indicator. And lastly, redox titration. In this type of titration, a reducing agent is titrated with an oxidizing agent or vice versa. The endpoint is usually detected using a redox indicator or by monitoring the change in oxidation reduction potential.
Assalamualaikum. My name is Iwani Tahira binti Ismail. So here I'm going to talk about the chemical and apparatus that we use to conduct this experiment. In this experiment, we use several chemical and apparatus including volumetric flask, conical flask, pipette, burette, method stand, natrium hydroxide solution, phenolphthalein indicator, and vinegar.
Assalamualaikum and hi. My name is Farah Patricia binti Azrul from class RAS 1204J, and I will talk about the methodology of this experiment. For the procedure, there is preparation of sodium hydroxide solution and standardization of sodium hydroxide. The first step is the mass needed to prepare 250 ml of 0.1 mole of sodium hydroxide solution was calculated. Secondly, all the sodium hydroxide was transferred completely into a beaker. About 50 ml of distilled water was added and slowly stirred until a sodium hydroxide solution has dissolved. Using a filter funnel and glass rod, the solution was transferred carefully into 250 ml volumetric flask. The beaker was rinsed a few times and the water was transferred into the volumetric flask. And the fourth step is the solution was transferred into a clean empty reagent bottle. And the fifth step is the burette was filled with sodium hydroxide solution. Next, 20 ml of the hydrochloric acid was pipetted into 250 ml conical flask. 2 to 3 drops of phenolphthalein was added as indicator. Next one is the HCl solution in the conical flask was titrated with the sodium hydroxide from the burette slowly while shaking the flask until the indicator color change. And the next step is the color change and reading at the end point of titration was recorded. I will talk about the procedure of determination of acetic acid in vinegar. Firstly, 10 ml of the given vinegar jalin was pipetted and diluted to mug with distilled water in a 100 ml volumetric flask. Secondly, 20 ml of this diluted vinegar solution was pipetted into a 250 ml conical flask. 2 to 3 drops of phenolphthalein was added as indicated. Thirdly, The vinegar in the conical flask was titrated with the sodium hydroxide from the burette slowly while shaking the flask until the indicated colors change. Next, steps 2 and 3 was repeated 2 more times, and then reading at the end point of the titration was recorded. Last but not least, the weight over volume percentage of acetic acid in the vinegar sample was calculated.
Assalamualaikum and hi everyone. My name is Ina Sofia binti Mahadi. In this part, I will present the result for percent content of acetic acid in vinegar by titration, which is one of application in titration that we choose in this topic. The data in this application consists of the standardization of sodium hydroxide solution and the determination of acetic acid in vinegar. So, this is the data for table 1, which consists of standardizing sodium hydroxide solution. The average volume of sodium hydroxide is 35.6 ml that will be used to determine acetic acid in vinegar. Next, for the data of table 2, show that the values of titration in order to determine acetic acid in vinegar. In this titration, we use 10 ml of vinegar in each titration. After analyzing the result obtained, the average amount of sodium hydroxide that we use is 9.25 ml. Next is the calculation to determine the percent of acetic acid in vinegar. The total acetic acid in vinegar is 0.6%.
To conclude, we managed to achieve the objective of the experiment, which is in this experiment to successfully determine the content percent of acetic acid in vinegar by titration method. This experiment serves as both physical and educational purpose. The meaning of physical purpose is this titration refers to the application of theoretical knowledge of chemical and analytical in real-life substance, which is in vinegar. Besides, by the meaning of educational purpose, are the experiments expose students to skill development, precise measurement, handling apparatus correctly and safely, and also applying mathematical calculation to real-life scenarios.